This week's Pulse sits at the edge of biology and computation, where tiny brain-like cultures are starting to learn simple patterns, electrodes are getting smarter, and ethics is catching up so the work can move forward without crossing lines. A lab reported a brain-like organoid that changed its activity after repeated training pulses and then held that change for hours, a basic sign of memory called plasticity. It matters because reliable, repeatable plasticity is the first rung on the ladder from curiosity to controlled biocomputing. Another group advanced the interface itself by packing thousands of soft, three-dimensional microelectrodes around and beneath an organoid, so the system can both read spikes and deliver targeted stimulation. It matters because better input and output means less guesswork, cleaner training, and a higher chance that results hold across multiple samples. A bioethics body updated guidance for organoid and biocomputing research with clearer donor consent language, stricter rules for stimulation regimes, and checkpoints for stopping experiments that show unwanted complexity. It matters because transparent guardrails reduce uncertainty for funders and oversight boards, which in practice speeds up well-designed projects. An organoid is a tiny, self-organizing 3D cell culture grown from stem cells that mimics some features of a full organ, in this case, the brain. Think of biocomputing with neural organoids as using living circuits to spot patterns, where neurons fire in spikes, and rewire their connections when signals repeat, which lets the tissue tune itself at very low power. The cause and effect is straightforward. When inputs arrive in the right rhythm, pathways strengthen and responses become easier to trigger, turning a noisy mass of cells into a trained filter. The near-term appeal is energy. A cluster of a few 100,000 neurons can respond to patterns using microwatts to milliwatts, while digital hardware doing similar work may draw watts to tens of watts, which is a 1,000-fold gap in some cases. The research value is insight into disease because these living networks reveal how genes, drugs, and stress alter learning in real time, which helps explain conditions like epilepsy or developmental disorders without invasive tests. The practical limits are real. Organoids vary from batch to batch, training is slow, and long-term stability still drifts, so any claim of brain-like intelligence should be taken as a narrow, measurable behavior, not a broad ability. Here is the clean explainer that ties those pieces together. In a typical setup, a high-density array senses spikes at thousands of sites, while a controller delivers short electrical patterns and occasionally a chemical reward. Then the array looks for a change in timing or firing rate that signals learning. Separating sensing and stimulation with more spatial precision reduces crosstalk, so you get better signal-to-noise and can train specific microcircuits rather than the whole blob, which is how you move from luck to control. To see why the energy claim matters, imagine using an organoid as a front-end filter that grades whether an incoming signal looks like a known rhythm before passing it to digital logic. If it rejects 90% of the junk at a power cost that is almost free, your downstream system gets faster and cooler. For disease work, the same setup lets researchers compare how different patient-derived organoids learn under the same training, which can reveal how a mutation changes synapses, or how a drug rescues that behavior, a tight feedback loop that classic cell cultures struggle to match. The limits define the engineering plan. Biological networks excel at fuzzy pattern separation, but are poor at exact arithmetic. So the right role is as a coprocessor that handles recognition, anomaly detection, or signal cleanup, while deterministic chips do counting and control. Fabrication and handling add friction because organoids need sterile conditions, temperature control, and nutrient flow, which means your computer lives inside a small bioreactor, and every pipeline must tolerate growth and drift. Repeatability remains the hardest problem. Two organoids grown the same way will not be identical, so you design for populations and statistics, not single devices. And you build training protocols that converge despite noise. All of this flows into a simple trend teardown. The driver is bio-hardware efficiency, because neurons compute with spikes and adapt their wiring on the fly, giving rich pattern processing at energy levels silicon struggles to touch. The trade-off is variability and scalability, since living tissue is never perfectly uniform, and scaling production from tens to thousands of units with the same behavior is an unsolved manufacturing challenge. The constraint is ethics and reproducibility, because donor consent, privacy, and the possibility of unwanted levels of complexity require monitoring, clear stopping rules, and shared protocols that other labs can reproduce. A grounded rule of thumb follows from that. Use organoid biocomputing when you need ultra-low power pattern recognition or disease modeling with plasticity in the loop, and avoid it when the task requires exact repeatability, durable 24/7 uptime, or strict arithmetic precision. If you are a builder, start with a hybrid architecture where an organoid handles a well-bounded recognition task and hands its verdict to a digital controller, then measure energy per decision and stability across weeks, not hours. If you are on the research side, define learning targets in advance with a small set of metrics like latency shift, spike correlation, and retention time, and commit to sharing protocols so other labs can confirm your curves. If you lead a program, budget for the interface first, because a better electrode array and cleaner fluidics often yield bigger wins than changing the cell recipe, and they make ethical review easier because they reduce stress on the tissue. If you focus on patients, tie every biocomputing claim to a clinical question, like whether a specific drug restores a specific learning curve, then publish the negative results alongside the positive ones to keep the field honest. The bigger picture is cautious optimism. We may never replace racks of GPUs with jars of neurons, and that is fine, because the winning path is likely a careful partnership, where biology does what biology is good at, and silicon does the rest. Carry 2 principles from this week. Choose biology for adaptive, low-energy pattern work you can measure, and choose silicon for speed, scale, and determinism you can guarantee. Subscribe for more Future Forge.